Hi everyone.
Hello.
Let's just wait for another, I think 2 more minutes, then we'll start.
I didn't think we finished chapter 14.
Really?
No.
Oh, okay. Chapter 14. Okay, so where are we?
No.
We didn't talk about it? Marybeth, turn on your audio. I cannot see anything.
Sorry about that. Go back a little bit.
Back? No, I think I talked about this. This is the penicillin. I remember I talked about it, that how we made,
Right. Okay, yes, I remember this slide.
And this is the same thing.
Okay.
And cephalosporin, I think I talked about that we had that 5 generation cephalosporin. No?
That doesn't look familiar.
Okay, so let me start then from here. But I think I did. Okay. Okay, so let's start. So next antibiotic, cephalosporin. Cephalosporin, we get it from the fungi, Cephalosporium acromonium. We have 5 generations cephalosporin. We modified 4 or 5 times. First generation, it was narrow spectrum, only on active against on gram-positive bacteria. Second generation, we made it extended spectrum. Okay, means with gram-positive, we added gram-negative bacteria. It should be uppercase. Third generation includes a Pseudomonas, again extended spectrum. Okay, mostly injected, few was oral. Then fourth generation, we made it completely broad spectrum, so we included all the organism, okay, both for gram positive and gram negative. Fifth generation, again it is extended spectrum, and we specifically made it for MRSA. Okay, now this group of antibiotic, they have beta-lactam ring. So basically they work on the cell wall formation of the bacteria. They attack the cell wall formation of the bacteria, means when bacteria make peptidoglycan. Okay, so they attack, they don't allow that NAM and NAG molecules to make that peptidoglycan. That's how that beta-lactam ring work. I hope you remember that beta-lactam ring. It looks like this. So that's the cephalosporin. Then vancomycin, they do not have beta-lactam, they have glycopeptide, special type of protein. Okay, and this group of antibiotic, we get it from the soil bacteria Actinomycetes. That vancomycin, especially we get it from Streptomyces virgini. Streptomyces, this is one type of actinomycetes. So they work on the inhibition of cell wall synthesis, large molecules that bind to the peptide chain of peptidoglycan subunits, blocking the transglycosylation. It's exactly same as how beta-lactam ring work on. Okay, so when they block that NAG and NAM to make peptidoglycan, that's the transglycosylation. Okay, and they also block the protein transpeptidation, mean from outside to inside. They don't allow the movement of protein, and that's the peptidation. They are bactericidal against gram-positive bacteria. They used as narrow spectrum, not active against gram-negative bacteria. Used to kill MRSA, but now bacteria got methicillin-resistant enzymes, methicillin-resistant protein they started making. Okay, so that it's not susceptible for all those MRSA. Now we are trying to make that that vancomycin resistant. Still, that also is not working. Bacitracin, the same as vancomycin, they have that glycopeptide. Okay, work on the cell wall composition of bacteria. We get it from Bacillus subtilis. Application is topical. We use it as ointment. Okay, now bacitracin, it is nephrotoxin. So if we only use bacitracin, it has bad effect on nervous system. Okay, nephrotoxic means it damages the nervous system, all the neurological new organ and brain, basically brain, the nervous system. So what we do to basically to decrease the effect of bacitracin, we add neomycin and polymyxin. So nowadays we make neosporin with bacitracin, neomycin, and polymyxin. So these three drugs together make the neosporin. Okay, and by doing this, we are actually reducing the strong effects of bacitracin. So it's not nephrotoxic anymore. It is a broad spectrum against gram-positive bacteria, Staphylococcus, Streptococcus for the skin, and a little bit of few of gram-negative bacteria. Block transport of peptidoglycan subunit across the membrane. So it does not allow that protein to move from outside to inside or inside to outside. So all those till up to this, bacitracin all worked on the cell wall synthesis. Now this group of bacteria, they work on the protein synthesis. Okay, they inhibit the protein synthesis mechanism of bacteria. So chloramphenicol, macrolides, and lincosamides, they bind to the 50S ribosomal subunit, prevent peptide bond formation, stop protein synthesis. Okay, so when they attached with the 50S ribosomal subunit, means the large one. Okay, what they do from A side to P side, when that oh gosh, that RNA, basically tRNA and messenger RNA combination, when they move from A to P, what they do, the tRNA, which one was carrying that amino acid, so it releases all the amino acids right over here on the P side. Now that 50S ribosome, subunit of ribosome, they actually make the peptide bond in between the amino acids, and that's how it makes a polypeptide chain. Now this group of antibiotic chloramphenicol macrolides, they don't allow to make that peptide bond in between, so all the amino acids remains loose. Okay, they are not interconnected with each other, means they cannot make a polypeptide chain, means they cannot make a protein. So that's how they stop protein synthesis. Aminoglycosides, they bind to the 30S ribosome, means the small subunit of ribosome, impair proofreading. So small subunit, they actually help to proofread, means okay, so when it is made, it is carrying mate, so over here that you know, the codon and anticodon combination should be exact same, that matches with the methionine amino acid. So if the proofreading machinery is not there, if they cannot read it correctly, so instead of methionine, if they bring glycine, okay, so then they actually targeting the protein synthesis, blocking the protein synthesis. So instead of insulin, if they make something else, let's say glucagon, then the purpose is not going to be fulfilled. Insulin, it actually decrease the sugar level. Glucagon, it increase the sugar level. Okay, so that's how this group of antibiotic targets to stop the protein synthesis, or it actually produce faulty proteins. Tetracycline bind to the 30S, means small subunit, block the binding of tRNAs. So over here, tRNA cannot sit on the messenger RNA. And then protein synthesis is not even, they cannot even start the protein synthesis. Aminoglycosides, this group of antibiotic like streptomycin, gentamicin, neomycin, they're bactericidal, broad spectrum, they're nephrotoxic, neurotoxic, and ototoxic. That was a nephrotoxic, means that was for the kidney. I read neurotoxic. So this affects the nephron of the kidneys. Nephrotoxic means it damages the kidney. Okay, this is for the kidney. Neuro means this is the nervous system, and ototoxic means it actually has effect on ER. All the function of all these organs are going to be damaged by overusage of streptomycin, gentamicin, and neomycin. Overusage or overdosage. Okay. Tetracycline, they are bacteriostatic, natural from Streptomyces, soil bacteria, and the semi-synthetic form, you know, we converted the tetracycline into doxycycline. Although the tetracyclines have broad spectrum in their coverage of bacterial pathogen, side effects that can limit their use include phototoxicity, eyes become too sensitive to light, permanent discoloration of teeth, liver toxicity with high doses or in patients with kidney impairment. So, and that's the reason, because of all these bad side effects that affects the body dangerously, we made semi-synthetic form of tetracycline basically, and that is the doxycycline. So basically we tried to lower of all those things. Okay, and that's the purpose of this semi-synthetic form of tetracycline. Macrolides, like example erythromycin, they are broad spectrum, natural from Streptomyces group. Semi-synthetic form of erythromycin is azithromycin. We still use azithromycin, and they are bacteriostatic. So whenever you see that there is any semi-synthetic form means either we are trying to reduce the dose or the effect, okay, or it might have lots of side effects. To control all the side effects, um, or to extend the, like from narrow to broad. So we want to include the few more groups of bacteria. Okay, so that's the purpose of making semi-synthetic form. Chloramphenicol, they are broad spectrum, and this is the bacteria first one to approved by the FDA. Natural from Streptomyces venezuelae, bacteriostatic. It has serious side effects like anemia, suppression of bone marrow production. Okay, bone marrow production is not there, then you can understand that we are going to get anemia, because all the blood cells like RBC, WBC, platelets, everything is going to produce from the bone marrow. Restricted, and that's the reason we have restricted usage on in USA. Nowadays, we really do not use chloramphenicol on that way, or maybe very low dose we use. Antifungal drugs target the different cell structure, so they work on the protein synthesis, on the DNA RNA structure, disrupt the microtubule function. Okay, microtubule transport everything inside the cell. So when not bacteria, like fungi is going to make protein, let's say this is the Golgi bodies, when the protein is prepared over here, then that protein needs to transport for the perfection, or you can say the packaging and all those things. So that is going to sorry, that this is, let's say this is the ribosome, and let's say over here this is the Golgi. Okay, so a protein is made over here, and for the packaging and distribution, everything for that protein needs to come over here. Now from here to here, that transportation is actually done by the microtubules. So if they don't do that, then protein is not actually going to be in work, basically. Okay, they are not functional protein, and all the cell, different areas of the cells are not going to get that protein. Not only that, that microtubule during cell division, they make that fiber that actually that from the centriole, that actually basically they help in the during the cell division. So all those things are going to be blocked. These antifungal drugs, they work on the cell wall of fungi. And they inhibit the synthesis of glucans. They are actually the component of the cell wall. So basically, cell wall composition and the protein synthesis and cellular structure, they attack. One of the antifungal medicine is Pollen's, nystatin. Okay, topical for yeast infection. Amphotericin B, systemic, we use it for systemic fungal infection. Inhibition of ergosterol synthesis, fungicidal, nephrotoxic. So that day, no, I didn't talk in this class. So ergosterol. So we have cholesterol. At the beginning, I think when I start this chapter, I talk about that. We have cholesterol. That's the type of sterol we have. Ergosterol is very specific type of sterol inside the fungal body. Okay, fungi, they have ergosterol. This is sterol, and remember the sterol, they are a very important part for the stabilization of the cell wall. Okay, if we can attack the sterol. Okay, then we can actually indirectly attack the plasma membrane. So we can dismantle the plasma membrane structure. So and the composition is completely different, cholesterol and ergosterol. So that's why we easily can use these antifungal drugs on our body. Now that Pauline's group, they have nephrotoxic effect, means they are very harmful for the kidney. Imidazole synthetic fungicide for ringworm specific for Tinea pedis, Tinea solium. Fluconazole, we use this for systemic yeast infection. Now the antiviral. Nucleoside analogs inhibit DNA synthesis. So for antiviral drugs, for virus, against virus, it was very difficult for us because virus, you can understand that they are obligate intracellular organism, right? So means they must have a living cell for their growth or development. Okay, so when we are going to targeting a virus, so what virus is going to do? Virus is going to be inside the cell, and what they are going to do, they are going to use our machinery to make the viral protein. So when, let's say, it is a human virus, okay, so when they are attacking human cell, they are using our enzyme, our machinery, all of the proteins, all of the enzyme, they are all of ours stuff. Virus is going to use to make viral protein. Okay, so what is happening when we are going to target that virus to kill? So basically we are targeting all of our enzymes and proteins, right? So it is very difficult to make that antiviral drug. It was not easy like fungus. Okay, at least there is one dissimilarities that they have for the fungi. They have ergosterol and we have cholesterol. So we can target over there. But for the virus, it's very difficult because they're using all of our machinery, all of our enzymes, all of our proteins, because they do not have anything except that either DNA or RNA. Okay, once they are in, immediately they get muted. They get control over our system. So and then it's very difficult to target the DNA and RNA. Okay, so we really do not have that many antiviral drugs in the market. We have one, Iciclovir, like Zovirax and all those we use, but in this case, so you can understand we have only option to target the DNA and RNA synthesis for the virus, viral DNA, viral RNA. Now the problem is, as I told, as soon as that viral DNA and RNA enter inside the host cells, immediately, within a fraction of a second, they take control over the host cell. Okay, and they use all the proteins enzyme to multiply their DNA or RNA. So what we usually do for these Zovirax and Acyclovir, so what we do, we cannot even kill or stop the DNA synthesis directly. Okay, because they are just using their template strand, but all the protein enzyme is working that are actually ours, that came from the host cell. So what we can do nowadays. So let's say it is adenine, guanine, cytosine, thymine. If it is DNA, these are the nucleotide, right? Same nucleotide virus is using. So what we do, we directly cannot target cytosine, we directly cannot target thymine, we directly cannot target any of the enzyme and the proteins. But what we do, we actually nowadays we make one special type of guanine. It's not guanine, but to trick the polymerase enzyme, and that is also ours, by the way, that polymerase enzyme. But we trick our enzyme. Okay, instead of guanine, it actually pick up that similar type of guanine that we make. Okay, and if it is called guanosine. So similar structure, almost similar structure. So when polymerase pick up guanine instead of guanosine, like let's say over here it is cytosine, and over here we must have guanine, but instead of guanine, if they pick up guanosine, because they look exactly same, they behave exactly same way. Then what happened, that cytosine and guanine, they really cannot make that hydrogen bond. They need 3 hydrogen bond to make. They cannot do that. So basically we make guanine analog, okay, and that's how we are trying to control the virus. Okay, that's the way the antiviral drug actually works. So that Zovirax we use against the herpes virus, selective inhibition of herpes virus replication, acyclovir conversion to nucleotide analog only in virus infected cells, and this only work on virus infected cell because you can understand all other cells are our cell, okay, and we cannot allow our own cell to make that that nucleotide analog, guanosine analog. So very little harm to uninfected cells, because anyway our target is only the virus infected cells. So acyclovir is a structural analog of guanosine. It is specifically activated by the viral enzyme thymidine kinase. It becomes activated when that enzyme is there inside the virus, when that virus comes with that DNA or RNA inside our body, and they have that, they carry very few enzymes. So one of the enzymes is kinase. If they have that thymidine kinase, then only we can do that. And then preferentially binds to viral DNA polymerase, leading to chain termination during DNA replication, okay? Because they cannot make that triple bond, and they cannot extend the DNA strand. Antiviral drugs for treating HIV/AIDS, because it can easily mutate the virus to become drug resistant. HIV is typically treated with a combination of several antiretroviral drugs, because they have that enzyme, okay, which may include reverse transcriptase inhibitors. That's the enzyme they have, reverse transcriptase, okay. Means that enzyme help them to make DNA from RNA, okay. So if we can stop the function of that enzyme, okay, that's the reverse transcriptase inhibitors. To do that, just one drug is not enough. We actually have a combination of different drug. So reverse transcriptase, when they do that they make DNA from RNA, means basically when they make the virus, there's a whole process. There are so many other enzymes are there, like protease, integrase. So we actually block all these. So first, if we attack reverse transcriptase, then they cannot make that one. If we then immediately, if we attack the protease enzyme, then like virus, they can make viral protein, they can make viral DNA, RNA, but they cannot. That if protease enzyme is not working, then they cannot make the mature virus. Now, if we do the integrase, if we attack the integrase enzyme, that integrase actually helps viral RNA to be incorporated inside the human DNA. Okay, so maybe let's say this part is the DNA. So that linking between the human DNA and the viral RNA, okay, so that joining is going to be done by the integrase enzyme. So if we inhibit the integrase, then viral RNA cannot be joined inside the human DNA. Okay, they cannot be part of the human DNA. If they cannot be part of the human DNA, then they cannot make the viral DNA, because that's what they do from RNA to DNA. Integrase does that, then that reverse transcriptase, it actually helps to first they make RNA and then they make DNA. So those things is going to be inhibited, and that's how we actually work on the antiviral drugs. So the drug resistance, why bacteria get drug resistance, or how they get drug resistant. So several important factors can accelerate the evolution of drug resistance, like overuse and misuse of antimicrobial product. Okay, the person, like let's say every month that person is going to get that like flu-like things, like cold and flu. So and every month that person is going to use that erythromycin to treat that that nasal congestions, to treat the fever and cold and cough and all those. After a certain period of time, the body will be resistant, means the bacteria inside the body will be resistant to erythromycin. So that's the overuse. Okay, misuse. The doctor has asked you to take the medicines for 10 days. Okay, and at specific time. Okay, so if you first day, if you take it at 7 a.m. in the morning, next day you have to take it 7 a.m. in the morning, or maybe every 8 hours or 12 hours, maybe in the evening you have to take exactly on 7 p.m. So if you, instead of 7 p.m., if you take it on, like, let's say, oh, I forgot, so it's 10 p.m., let me take that. So bacteria is going to get 3 hours period over here. So 7 to 10. By that time, they will be active. Okay. So, and they will multiply. Not only that, they will be resistant also. Okay. So all those things are using, instead of say 10 days, if you continue only 5 days, after 5 days you feel good and you stop taking. What does it mean? It's 10 days is required to kill all the bacteria. So instead of 10 days, if you take 5 days, that means your body is still carrying 50% of the bacteria. Okay, and all those bacteria is going to be resistant, because remember, the bacteria, they do transformation, they do horizontal gene transfer. So after 5 days, what happens? 50% of bacteria is dead, and 50% of bacteria is alive. Right now, all this 50% dead bacteria, they are going to release their chromosome, And these 50%, they are going to pick up all those plasmid, and they will be resistant to that drug. Okay, because they can do that horizontal gene transfer. They can pick up the genes from the environment. As soon as they are going to pick up these genes, immediately their body is going to make antibody like a structure, and that will make them resistant. Means they know how to protect that antigen. Over here, they know that they will come to know the structure of that antibiotic. They didn't experience that antibiotic, but from the gene, they are going to learn the structure, and they will make that anti-protein against that thing. And that's basically, to do that, they are going to develop antibiotic resistant genes inside their plasmid, and they will transform into mutant bacteria, and that's how that drug resistance occurs. So penicillin G resistance of Staphylococcus aureus from 3%, now it's 200%. Okay. Multi-drug resistant Staphylococcus aureus, MRSA, even BRSA is not also, we use that BRE, it does not work. Multi-drug resistant, like for the TB, MDR TB, that's also got resistant. So we now tuberculosis, it was out of the world, whole world. There was, I think, 2 or 3 years, consecutive years were there where the whole world didn't experience any, there was not even a single case of TB. Now it is re-emerging. Now it's re-emerging disease. It's coming back because of the drug resistance. Okay, and you can understand that horizontal evolution for the gene transfer, and vertical evolution is there. When one bacteria is multiplying into 2, they are going to get that drug resistant capacity. A variety of mutations can lead to antibiotic resistance, and now bacteria know all the different type of tricks. What they do, they know how to inactivate the drug. They know how to prevent the penetration of the drug. Alteration of drugs target site. When they're like penicillin, it is targeting the cell wall formation, right? When it is targeting cell wall formation, okay, so that penicillin has to reach over there. They don't allow the antibiotic to go over there. They modify the target. It looks like that, like how we actually, for antiviral, how we made guanosine instead of guanine. Exactly same trick. Actually, we learned the trick from the virus, bacteria. What they do, it looks like double layer, double lipid layer, phospholipid layer, but it's not. Instead of sitting over here, the structure looks similar, that it's a phospholipid. Okay, so penicillin sits over there and nothing can happen. Rapid ejection, okay, they know how to throw out all the antibiotic outside of their body, that efflux. So these are the procedure they follow. So efflux pump, what they do when antibiotic reaches inside the bacterial cell, what they do, they modify the plasma membrane structure, okay, so that that part of the plasma membrane or cell wall is very transparent for this antibiotic. Okay, it becomes very easier for this antibiotic to go out, because there is almost like a whole pore like structure. Okay, and it goes out. Inactivation of enzyme like penicillin. Okay, when it was penicillin, bacteria, what they did, they make penicillinase. Okay, that destroy all the protein of the all the enzyme of that penicillin. Now they can make methicillinase. So when they know the structure, they can change because of the mutation. They can make all these. So that's how they, they inactivate penicillin, and that's how the penicillin is not working. Target modification, as I told, that it is that enzyme supposed to sit over here on 30S ribosome. Bacteria is not allowing that. Block the penetration, even that absorbent capacity of the bacterial cell wall. They increased the absorbent capacity. So, or maybe they changed the glycoprotein receptors over there. If two ways that the antibiotic goes inside the bacterial cell, it could be that receptor attachment. Okay, it could be that. If they change this target, then it cannot sit over here. Okay, or maybe through the diffusion. So absorbent capacity, if it increases, then they cannot move through. Okay, so all these are the different procedure bacteria use, and that's how all these drugs they are resistant. Different bacteria actually works in different way. So this is the antibiotic, if they alter the target molecule. So this need to sit properly. Shape should be perfect. Otherwise, it cannot go inside, or it cannot go to target on DNA or RNA. So they do that. Efflux, they throw it out, blocking entry, and inactivate enzyme. Okay, so these are the 4 ways bacteria get resistant. So misuse of antibiotics selects for resistance mutants. Misuse includes using outdated or weakened antibiotics, using antibiotics for the common cold and other inappropriate conditions. You have a viral infection and you are taking that antibiotic. Okay, you are not supposed to take that because it's viral disease. When one antibiotic is inside your body, all the good bacteria, they get used to, and they become resistant, and from there all others. So it's inside your body. You are thinking, okay, so it's only me. No. So E. coli, staph epi from your body, it is going to your partner's body who is living inside your house. Everybody is going to get. Not only that, when you are throwing all the trash, the people who are actually collecting all the trash and they are dumping somewhere over there, all the bacteria. So the whole area is going to, whole world is going to be mutated, all the bacteria. When you come in the school, okay, you are eating something, or somehow you throw something inside the trash, we all are going to be infected. So no, you are not the alone is going to be antibiotic resistant. No, we all are. So it has a tremendous effect. Using antibiotics in animal feed, that's another problem. Like when you are drinking milk, all the cows, for overproduction of the milk, with their food, they are taking antibiotic. Failing complete the prescribed regimen, using someone else's leftover prescription. So all these are, what we do, it's coming from our, misuse, and we should not do all these things. The end. Okay, so this chapter is done. Do you have any question?
No, ma'am.
Nope. Okay, let me then go back to chapter, which one we are going to use? Chapter 15.
This is Nandi.
Mhm.
I noticed that our for chapter 15, chapter 14, we don't have the chapter on the online text on D12. I didn't see the chapter 14 text.
Oh, yeah, I saw the text. I will check. I don't. Okay, I will try to upload. Do you need that, or you can go to that Openstax?
I'm talking about open, or like you mean to go to Openstack itself?
Mm-hmm.
Okay.
I don't know if I can upload from my home. I will try. If I can,
Okay.
I will upload. Okay.
Okay.
Yeah. Okay. So chapter 15. Chapter 15. Okay. Slideshow. Okay. So chapter 15, it talks about microbial mechanisms of pathogenicity. How one microbes become pathogen and what they do, what are the different factors they use to become pathogen actually. So the disease, any condition in which the normal structure and function of body is impaired, or you can say damage. Okay, that is called disease. So any deviation from a condition of good health and well-being is called disease. So when you have, let's say, broken leg, is that a disease? Because that's a deviation from a condition of good health. Okay, your normal structure and function of body is impaired. You cannot walk. Okay, and maybe for the broken leg you have pain and all those things, but still it's not considered as a disease. It is an accident. Okay. Infectious disease condition caused by the presence or growth of infectious microorganisms or parasite. So to have disease, you must have infection inside of your body. Infection means successful colonization of microorganism in a host body. So one host, they have to enter inside your body. Okay, they have to enter. That's the first thing. Then second thing, they have to overcome your immune system protection. Okay, and once they overcome the immune system, then they have to start multiplying. They have to have that machinery. If they cannot multiply inside the body, it does not make any sense. Okay, so they start multiplying. Once they multiply, they make new colony. That means they can establish an infection inside your body. That's the infection. So when you tell that, okay, so I have an infection inside my body, that means you have a colony of microorganism inside a body. It's not just 3 or 4 cells, or maybe 300 cells entered inside your body. That it could be 3 cells, it could be 300 cells. If they cannot make colony, then they cannot establish infection inside your body. Okay, so they have to have colony inside your body, and that makes the infection. Okay, So infectious disease, if you have infection inside your body, means you have infectious disease inside your body. So pathogen, anybody that can cause disease, that can make colony inside your body, they are the pathogen. Non-infectious disease non-infectious disease means you are sick, you have disease. Okay, it's not your normal regular structure of your body, and you cannot do all the regular activity, regular function what you used to do. Okay, but there is no infection, there is no pathogen. Pathogens are not responsible for that deviation from the normal condition of your good health. Okay, so what are the reasons? One could be, so we are talking about non-infectious disease. One reason could be hereditary. Heredity, like let's say sickle cell anemia. So you are going to get it from your parents. You inherit the gene, sickle cell gene, from one of the parents, and that's how you get that disease. Okay, in that case, what happened, there is no bacteria or no pathogen is involved, but it's a disease. Okay, then let's say immune deficiency. Okay, immune deficiency, you get allergy. So dysfunction of immune system cells, like that your white blood cell immune system cells, mostly the WBCs, they can recognize the self protein versus foreign protein. Okay, so if self protein means it's your own cell, when they cannot differentiate between self and foreign protein, they actually, how they attack the foreign cell, they actually attack your own cell, and that's how you get the allergy. Okay, so dysfunctional work of immune system. Okay, you can have acquired immunodeficiency disease, means like let's say AIDS, HIV. So for the AIDS, it started with HIV. Slowly, it actually, disabling the immune system. It is making poor. Because of this disease, slowly your immune system is not strong. It's not working like before. So when you are at this stage, AIDS, you actually acquired that, poor immune system. So that's a non-infectious disease. Third, let's say environmental. Okay, so from environment you can get now. This is the time for pollen. So you are sick. It looks like flu-like, symptoms, but it's not flu. It's for the pollen. Those are the allergen that induce your immune system to become active, because your immune system can recognize that these are the foreign protein, so they become very, very active. Okay, so when they're active, they secrete so many different type of chemicals, and that's the reason. Okay, now nutrient deficiency, you can say lack of nutrients, like let's say scurvy. When you get scurvy, means you have you are vitamin C deficient. Your body is not getting enough vitamin C. So for all these 4 cases, you know what? No pathogen is there, but you are sick, you have disease. Okay, so the difference between infectious disease and non-infectious disease is, for infectious disease you have to have an infection, or you can say the infectious disease pathogens are responsible. You can say that pathogens are responsible. Okay, but over here there is no pathogen. Okay, so that's the difference between infectious and non-infectious disease. Sign. So objective and measurable evidence of disease as noted by an observer. You as a nurse, doctor, as a parent, you can see the signs, like a person is coughing, okay, person has fever. You can measure the heart rate, you can measure the blood pressure, you can measure the breathing rate, you can see the rash. So all these are measurable and objective. Anybody can see all those things. When as a nurse, when you are checking, let's say you are observing the stool report of any patient, okay, and you can, inside the stool you can see the presence of RBC. Okay, that's the sign. You can see that RBC is not supposed to be there, but it's there. Blood is there. That's abnormal, so that's the sign. Versus symptom, it's subjective evidence of disease and only experienced by the patient. Okay, so it is pain, it is paining. Can you feel it? No, only patient can feel. Nausea, headache, fatigue, chills. All these are only going to be evidenced, or it only going to be felt by the patient. Okay, so that's the symptom. Symptom, collections of signs and symptoms often associated with a particular disease, and that's why they are the syndrome. Okay, like you can say, let's say toxic or septic, toxic shock syndrome. In this case, lots of signs and symptoms are there. Fever, rash, you can say what you call skin peeling, okay, then low blood pressure. Okay, so all are together represent, when all these combinations of signs and symptoms are there, that indicate, okay, so this could be toxic shock syndrome. By seeing this combination of signs and symptoms, we can identify the disease, and that's the syndrome. Asymptomatic or subclinical disease that that do not show any noticeable signs or symptoms. There is no signs, noticeable signs or symptoms, like herpes simplex virus, like for varicella zoster, you can say, for the pox, for at this moment for the shingles. Okay, for long time that varicella zoster virus, it's inside your body. Okay, but you don't have any signs or symptoms. After 30-40 years you can start getting all the signs and symptoms, and it can develop shingles. Okay, so during the 30 or 60 30 to 40 years of your life, it was there, that virus was inside of your body, somewhere inside your body, but you didn't know that. Okay, so that's the asymptomatic subclinical disease. Exactly same thing happened for herpes simplex virus HSV1, and there are two versions, HSV2. This is for the oral herpes, means infection, you can say surrounding your surrounding the mouth, lip, you get cold sores. Okay, you can get it from kissing, you can get it from using all the utensils. If you use the same glass to drink water, you are going to get that. HSV2, you are going to get sores around the genitalia, you can get around the rectum. Okay, if you have HSV1, then after certain years you are going, they actually going to convert into HSV2. You are going to, chances are there that you are going to get that. So these are the asymptomatic. Now what we do to identify a particular disease, we actually rely on the signs and symptoms, and for for the identification, many syndromes are named using the nomenclature, special type of nomenclature. Okay, so and they are named by using that actually indicates the sign. Sometimes it's signs, sometimes it's symptoms, sometimes it indicates the location of the disease. Okay, and that's how we actually give the name of the disease, the syndrome. So the suffix emia means presence of an infectious agent. When it is viremia, means presence of infectious virus inside your body. When it is bacteremia, presence of bacteria inside your body. When it is toxemia, means you have toxin, special type of toxin. It could be bacterial, most of the time bacterial toxin, or any parasitic toxin present inside your blood. When it is septicemia, means presence of an infectious agent in the bloodstream. When that bacteria is present inside the blood, that is septicemia. When bacteria present inside your body other than the blood, that's the bacteremia. That's the difference between bacteremia, septicemia. Itis means inflammation. So pharyngitis. Okay, this by this name one can understand, okay, what happens, what's the symptoms or signs. It's the inflammation. And what's the location? It's pharynx. Lysis, destruction, like destruction of the red blood cell, hemolysis. Pathy meaning disease. Neuropathy means that affecting nervous system. Oma means tumor. Lymphoma means the tumor on the lymphatic cells. Classification of diseases. How many different types of diseases are there? It's a communicable disease, capable of being spread from person to person through direct or indirect contact. What is direct contact? Like for the, let's say it's coronavirus at this moment. Okay, COVID-19. That's the name of the disease. What is the direct contact? The person is sneezing, coughing, okay, and the other person, when that person is picking up that air drops, okay, means with that air drops, the person is picking up the virus. So that's the direct contact. Like for the nurse who is treating, nurse is measuring the high blood pressure, measuring the blood pressure of the patient, and patient is coughing or sneezing. And that nurse is breathing from the same air. So through the nose, she or he is going to get that virus. So that's the direct contact. Indirect contacts, person, like from the fomite, any fomite. So it's the negligence of nurse. She was, measuring the temperature of a coronavirus patient. Okay, and another patient, he went for heartburn. He didn't have that coronavirus. That nurse forgot to change the cap of the thermometer. What can happen? Okay, so from that cap, this person is going to get the virus, because that cap is carrying that saliva of this person, saliva associated with the virus. Okay, so this person, it's not for the direct contact. This person maybe didn't see even this person. So this is the indirect contacts. Okay, so let's say there is a sink in the patient's room, and over there is coronavirus, because that patient actually washed her mouth. Okay, now that patient is gone. Where did he or she went? I don't know, but maybe she died, or maybe she go back to home, but she left all the viruses over there inside the sink. Now next patient, she is not COVID patient. Okay, she is maybe heart patient. So he is here. Okay, and he is not feeling good while he was washing his mouth or face. Okay, and when he was not feeling good, he kept his hand over here on the sink, maybe to get the, on his hand he put all of his weight, because he was not well enough to stand still or stand straight. So he needed support, so he placed his fingers, his palms over here. Okay, and he put all of his weight over there. Okay, and after 15-20 days, that person can get that coronavirus, COVID-19. So that's the indirect contacts. Contagious disease, easily spread from person to person, like measles. Okay, like this person, if you are inside the same room, you don't even have to touch, you don't have to be closer. Like for coronavirus, if you maintain 6 feet distance, okay, you are fine enough, because you are not breathing from the same air. Over here, if you even that 6 feet, 10 feet, 20 feet does not matter. If you inhale the same air, you are going to get that. Okay, if you touch the dry skin from that person, you are going to get. So in this case, it is easily spreadable. It's easily spread from person to person. Now, what's the difference between then communicable and contagious disease? Communicable disease capable of being spread from person to person. In this case, easily spread from person to person. Okay, so we can say that all contagious disease are actually communicable disease, but not all communicable diseases are contagious disease. Like let's say gonorrhea. I think I forgot the spelling. R-H-A-E, I guess. I forgot the name, spelling. Anyway, the gonorrhea. Anyway, so that gonorrhea, in that case, what happened? it is communicable. Okay, if you have very intimate contact with that person, you can get that. Okay, but so in that case, it is actually communicable disease, but it is not contagious. If you are living with that person within the same room, you are not going to get that unless you are going to have that intimate contact. Okay, so this is communicable but not contagious. But measles, if you are staying with that person inside the one room, you are going to get that disease. So all contagious disease are communicable disease, but not all communicable disease are contagious. Okay, so that's the difference between these two. Iatrogenic disease occur after a medical procedure. Medical procedure like let's say it could be you are there in the hospital for wound treatment. Okay, and they cleared the wound, everything they did, but you know after that treatment you got tetanus. Okay, so because when they stitched, okay, you have a big cut, so you got, let's say, 5 or 6 stitches. Okay, they used the needle that was not sterilized, and it was carrying that Clostridium, Clostridium tetani spore. It was there, it was infected with that. But because now for the coronavirus pandemic, they do not have enough people to sterilize all those things. They do not have enough procedure machinery to do the sterilizing thing and all those. That needle, for a long time people didn't use that needle. Okay, and Clostridium tetani, they made spore over there, and that can go inside your own treatment. So that happens because of that stitching procedure. Okay, so you had a medical procedure over there, and then you got that disease. You went for this and you got that. So that's the iatrogenic disease. Then it could be side effect of drugs. So you had something, okay, like for coronavirus, now people are taking that hydroxychloroquine. Okay, that hydroxychloroquine is not good for who has high blood pressure, who has high blood sugar. So because of that treatment, they are going to get something else. Okay, so that could be one iatrogenic disease. Contaminated instrument, I already told you, like let's say after radiation, or okay, so radiation or chemotherapy. So for radiation chemotherapy, what you can experience, you can have nausea, you can have hair loss, you can have what you call anemia, you can have low blood pressure. So all these things you get when you have that you get through all this, like radiation and chemotherapy treatment. Okay, what else? Like surgery. If you had surgery, let's say, okay, there could be some complication that can arise, because of the surgery. So all these are the example of iatrogenic disease. Nosocomial disease occurred in hospital settings. Okay, so what happened? So all these coronavirus patients are there. Okay, and they are using same ventilator. So now we do not have enough supply of ventilators. So 4 or 5 people are using that ventilator. Now, one of the sick patient, he or she has MRSA. She used to go to gym, so she is carrying MRSA, okay? And she got immune with MRSA, so she does not have any kind of sign and symptoms for that. But all other 4 or 5 people who are using that ventilator, they are going to get that MRSA. Okay, so that you picked up from the hospital. It can only happen if you are in the hospital. Like you can get that MRSA from the nurse. As I told, all of my nurse students, they carry this MRSA inside their body. They don't even know. Okay, but they got immune because every time they're inside the hospital, they're working with the patient, so they got immune. But a new patient, it takes time to be immune. It has bad, all those, things that person is going to get. So that's the nosocomial disease. Zoonotic with the animal vector. So vector means who actually carry the pathogen. Like for malaria, if you get a bite from Anopheles mosquito, the name of the mosquito is Anopheles. So many different types of mosquitoes are there, but you have to get bite from this Anopheles. Only this type of mosquito can carry that Plasmodium vivax malaria. That's the protozoa actually, Plasmodium vivax. So only Anopheles mosquito can carry. Okay, so if you get a bite from that mosquito and that mosquito is carrying that protozoa, then you are going to get malaria. Rocky Mountain spotted fever, if you get a tick bite, okay, then and the tick is carrying that what do you call rickettsia. Anyway, so what happened in this case, the tick and that mosquito, they are the vector. They don't have any disease. They are not infected with the pathogen. But they are going to infect the person whom they are going to bite. So that parasites that came from this animal, and that's why it is zoonotic. Zoo means animal. Non-communicable disease tetanus, legionnaires, like Clostridium tetani, I told you. That Clostridium tetani, so you only get if you have, there is a wound, any cut or wound, and that spore can go through that wound inside the, enter inside the blood circulatory system. Then only you are going to get, okay, because that spore for Clostridium tetani, blood entering the blood is the easiest way to go to the neural cell, nervous cells, because that's the area region they actually infect. Okay, if you ingest the spore, nothing will happen. Like anthrax bioweapons, okay, that anthrax, if you inhale, it's dangerous. The coronavirus, okay, so if you ingest coronavirus, like now they reside inside your throat, inside your pharynx, okay, if they enter inside your digestive system, you are fine. They cannot make the colony. They are not going to give you the infection, because that route is not a proper route for this virus. They cannot tolerate all the high acidity, that environment they cannot tolerate. And the peristalsis, long, expansion and contraction of the digestive tract, they cannot, that mechanical exertion, they cannot tolerate. Okay, but if you inhale, okay, if so how it goes, like this is your mouth, okay, so one part goes directly to the, this is your esophagus, digestive part, and from here there is your trachea, respiratory part. Oh my God, what happened? Okay, respiratory part. So instead of going over here, if they enter over there, that's the preferred pathway for them. So they can survive exactly same way that Clostridium tetani, they goes through the cut. Okay, now the person who has the tetanus, you can touch, you can drink from the same glass, nothing will happen because it has to go through that cut part. Any other part, cut through the wound, and any other way, they won't survive. So you are not going to get that disease from any infected person, and that is why this is non-communicable disease. How much time do we have? Okay, so types of non-infectious disease. There are different types of non-infectious disease. Non-infectious disease means we are not going to get from that infected person. Inherited genetic disease like sickle cell anemia. You have to inherit.
It's 12:19, almost 12:20.
Oh, what time we are going to stop this class?
12:15.
Oh, okay. So, we are out of time. Okay, so let's stop then. Oh, okay. So I will see you on Thursday.
Bye, Professor Connie.